Thank you so much for that kind invitation. Really like to thank the organizers, especially Professor David Lai, for having me today to share some of our work on the utility of metagenomics in the era of COVID-19. If not because of this pandemic, I wish I would be there with you in person. So I think the first thing that probably has been covered is why has there been such an explosion in interest in microbiome? And we know that for many decades, we've been scratching just the tip of the iceberg. And now with the arrival of molecular methods, high throughput sequencing, now to omics, from metagenomics to metabolomics, and of course, the crux is all in the system analysis. So it's quite exciting, I think, for all disciplines, because it's like the invention of the microscope. We are actually finding new things that we never knew existed before. And not only are we now being able to tackle the predominant population, which is the bacteria, but also the dark matters of the gut, going all the way from the virome to the fungi, even to the sort of parasites. So I would say we've entered a new era of the revolution of what microbiome medicine could really offer. Now, if you think about some of the techniques that we've been using, with metagenomics, it clearly allows us to study the microorganisms in more depth, just through a sample. Our group has been working mainly with fecal sample, clearly now moving also to oral, buccal swab, breast milk samples and urine samples. And essentially, it actually allows us to understand what is there, what they do, who their friends are, the functions as well. And the beauty is to bypass some of the need of all the conventional ways of culture techniques. In the past, a lot of groups have been using 16S sequencing, and many still do now, and that allows us to look at the sort of genus, sometimes the species, but certainly may not be able to allow us to understand the functional profiling of these microbes. Having said that, now with shotgun metagenomic, we can really look at the taxa, and within a single sample, also understand the phages, the bacterial phages within the tissue or the fecal sample. So the workflow of metagenomics, there are many standard operation procedures, from sample collection to DNA extraction to library preparation to sequencing, data processing, and then the taxonomy and the functional sort of profiling. In the past, we used to outsource, but right now we run in-house sequencing here at our microbiome center at the Salt Science Park. I think the bottom line, what's really important is how all these SOP, what you see here, every single step can really influence some of the results that you see, even with different types of DNA extractions or methods. So really, there's a huge call for standardization of what we really do. We started working on this about 5 years ago, and when the COVID-19 pandemic hit about 18 months ago, we realized that COVID-19 is not just a respiratory disease. The human gut is also a main target because the virus can be found in many parts of the human intestine. And the reason for this is the viral entry receptor, ACE2, is clearly highly expressed in the human gut. If you look at this chart here, we see that ACE2 is expressed in the lungs, in the kidneys, in the liver, in the vasculature, such as the blood vessels, but the abundance is huge and very enriched in the small bowel and the large bowel cells. And a landmark paper initially then started following patients and looked at different types of samples, ranging from the pharyngeal swabs, feces, sputum, blood, urine, and nasal swabs. And you could see this longitudinal cohort. At some point in time, every person has actually active virals detected. And live SARS-CoV-2 were found in the stool sample of 2 patients who did not have diarrhea. So from our perspective, then we started thinking, look, from even the GI physician point of view, it's clear that not all SARS-CoV-2 patients have GI symptoms, but even in these asymptomatic people, the detection of the virus in the stool is present. It's more so in the severe cases, and it does not always correlate with people having symptoms. So asymptomatic carrier state does occur. Then in the first set of the work we did short-gut metagenomic sequencing and look at the genome of the SARS-CoV-2. What we found is that we know that in vitro transcription analysis have shown from infected cell model that they express the 3N and the 5N of the SARS-CoV-2 genome. Now, with this higher 3N is an indicative signature of active viral replication of the infection. And it was rather alarming when we find that when the throat swab of the patient is negative, several of these patients continue to show high end of the 3 active replication in the stool sample, suggesting that even despite disease resolution, some activity is still present in the human gut of patients with COVID-19. Where does the gut microbiome come in then? So we started looking at the gut microbiome taxonomy of COVID-19 patients' stool samples, and we compared those who have very sort of high levels of SARS-CoV-2 virus. We see that they harbor much more opportunistic type infections and pathogens, and these are some that can cause secondary infections or bacteremia, and some of these bugs are derived from the oral cavity. In contrast, subject samples who actually were enriched with very low sort of SARS-CoV-2 infectivity, or sometimes none, they harbor more beneficial bacteria known to produce short-chain fatty acids. So in this earlier work that published in Gut, clearly some patients with COVID-19 had active and prolonged SARS-CoV-2 activity in the gut, even in the absence of GI manifestation. So the implications for public health at that time was clearly huge. It shows that the virus can be detected in the stool through metagenomic sequencing and through PCR, even after recovery. And sometimes it persists for several weeks after discharge. And in the Chinese Museum of Hong Kong, we started applying stool tests for SARS-CoV-2 PCR as an accurate and safe measure for certain groups of subjects, such as the infants, the toddlers, or a specific group of elderly people. And although this has not been proven, there's always the caution on the potential threat of the fecal-oral viral transmission. Coming to the gut microbiota, we know this is like the conductor of the immune orchestra. It regulates our immunity, it maintains defense against viral and bacterial infections by stimulating the appropriate regulatory immune cells defense. And in simple terms, when there is what we call an imbalance of these gut microbes, or so-called the terminology of these viruses, clearly it makes certain people more susceptible to infections, and some may have worse outcomes. So in the first paper that we published in Gastroenterology, that was in March last year during the early pandemic, we collected samples from patients with COVID-19. This was during the first wave of COVID-19 in Hong Kong in February to March last year, at 15 controls. And we collected samples every 2 to 3 days from admission until discharge, and we profiled the gut microbiome using metagenomic sequencing. Then there was a second and a third and a fourth wave in Hong Kong, which is unfortunate, but that allows us to therefore sample a larger cohort of COVID-19 cases, up to 100, with a varying degree of severity from mild, moderate, severe to critical, as well as a large pool of control subjects, 1,500 people who don't have COVID-19. And also we sample a positive control group of people with pneumonia, but negative for COVID-19. What we did was when we did shotgun metagenomic sequencing on these fecal samples, the most intriguing part is that amongst all the host factors, you can see that is known to affect the gut microbiome, from age to host factors, including antibiotic use, diabetes, comorbidities, COVID-19. The presence of having SARS-CoV-2 stands out as one that has the highest magnitude in an altered gut microbiome. And we found that the baseline gut microbiome of these patients also correlated with the fecal viral load of SARS-CoV-2. Essentially, the 4 Bacteroides species showed significant inverse correlation with the fecal SARS-CoV-2 load, and all these species were reported to downregulate ACE2 expression in the mutant colon. The next question we asked was, now, how does this affect the immune response? Some of these cytokine storms that we see in more severe patients who end up in intensive care unit. When we compare the mild, moderate, severe to critical group, we could see that they can be differentiated by this PCA sort of analysis according to clearly the inflammatory cytokine markers in their blood. But in addition to that, there were several gut commensals with known immunomodulatory functions that were underrepresented in COVID-19, and they were more depleted in those subjects who were more severely ill or more critically ill. For example, here you could see with Bacteria praxleuxi, an anti-inflammatory bacteria. And this anti-inflammatory bacteria and also short-chain acid producer was persistently depleted in COVID-19 patients, even after they have actually recovered from the disease. And in addition, these species that were depleted were also at the same time associated with increased inflammatory marker concentrations, such as in the plasma or the blood of these subjects with CXCL10 or even chemokines or certain cytokines. They have a sort of direct correlation with the severity as well as the bacterial composition. We then followed these patients up to 30 days after they have recovered and gone home. And here you could see the differentiation between the non-COVID-19 gut microbiome and the patients with COVID-19 who have recovered, and they are still completely well separated, indicating that their gut microbiome composition remained very, very different. There's also the very alarming phenomenon that up to 2/3 of patients may develop what's known as long COVID, which is post-acute COVID syndrome, even after recovery. This is seen in the United States, in Europe, in UK, as well as in Asia. And these symptoms in the post-acute phase can range from persistent lethargy to breathlessness to certain loss of memory or even some other respiratory and multi-system disorders. And what we found was that for some patients, up to a few months after recovery, they persistently have very low levels of the immunomodulatory bacteria, from the Eubacterium species, the Rosabaria, the fecal bacteria, Prosnutzii, and bifidobacteria. And whether this could certainly contribute to some of the symptoms that have been well described now. And we then followed these patients up to 6 months after they recovered. And here on the left, you could see that those who have post-acute COVID syndrome, the yellow ones, those with no post-acute syndrome at all, no symptoms at all after recovery, the blue, and then the gray are the non-COVID type controls, and they're well separated actually completely. And of all these over 100 patients, we found that there were actually quite a lot that have poor memory, anxiety, hair loss, mental disorder, difficulty in sleeping, as well as blurred vision. And they continue to have them up to at least 24 weeks. Now, when we then look at how different the gut microbiome is in those who had long COVID, you could see actually how they actually continue to persist in terms of composition of the gut microbiome when they're compared to some non-COVID-19 controls. And amongst all these multi-system symptoms, there are certain bacteria or composition based on this analysis that can categorize them into certain enterotypes. So when we look at GI symptoms, respiratory symptoms, more general symptoms like fatigue or muscular symptoms or neurological symptoms, you could see here that there were some that actually were able to predict some of these development of symptoms. What's quite interesting is that when we look back at the gut microbiota composition at baseline, which is at admission of these patients, with those with no long COVID and those here with COVID, we are able to find gut microbiome baseline composition that predicts if someone is going to develop long COVID or not based on their composition on admission. So therefore, I think what you could see here is that clearly for each of these different types of multi-systems, there's certain composition of the family of bacteria that were able to be associated with a very specific symptom. So here is a summary of some of the illustrations. In health, we well know that we all have certain bacteria that categorizes each uniquely like our signature. And they are here for some anti-inflammatory and so boost immunity purpose. But in the acute setting of COVID-19, there's certain depletion of this, and this depletion continued to be sort of prolonged or altered until after recovery. So the application of metagenomics in COVID-19 is quite important because apart from understanding what is there, it also allows us to know what they do. Then we started looking at what the functions are based on the microbial pathway and the metabolite. So we did targeted and non-targeted sampling. And we found that the microbial pathways that were certainly significantly reduced in subjects with COVID-19 were those related to short-chain fatty acid production and isoleucine biosynthesis. And again, with targeted metabolites, these are the ones that remain depleted in patients and were also associated with more severe disease with raised CXCL10 as well as C-reactive protein. So apart from some of our work, clearly from globally, there are already now 5 observational studies showing that COVID-19 patients had altered intestinal microbiome compared with controls. and they harbor increased numbers of opportunistic pathogen, some of these seen in the immunocompromised host. And again, studies have indicated that fecal respiratory microbiome dysbiosis was associated with increasing COVID-19 severity. The real question is, can we do something about this? For example, can we improve health by modulating the gut microbiome? Many things affect our gut microbiome, from what we do, what we take, whether we exercise, from the diet, from the drugs we take. So the risk factors for imbalanced gut microbiome are the elderly, people with comorbidities like diabetics, people who are obese, and those with comorbidities. And these group of patients happen to be on epidemiology work to be the ones, if they are infected with COVID-19, have worse outcomes as well. So that takes us to how the gut-lung crosstalk occurs. We know that clearly the gut bacteria produce certain metabolites that is then circulated through our blood system. And some of these can actually affect some of the respiratory functions as well. And certain immune activity against influenza common cold are also part of these bacterial species, such as NK T cell activity or T suppressor or T helper cells activity. So the possible mechanisms of these dysbiosis by SARS-CoV-2 virus in some of these susceptible individuals or those who have higher mortality may in part relate to certain depleted species that is well described in subjects with high comorbidities. So with that, some of our students then published a review article studying the literature on the use of probiotics, prebiotic, and dietary approaches during COVID-19. And on top of that, by using some big data analysis as well as machine learning, looking at some of the integrated analysis, we then at the Siew HK developed a unique microbiome immunity formula, SI101, based on some of these machine learning. And together with our Dean of Medicine, Francis Chien, we're able to sort of launch this in the field at the press conference, understanding how the citizens could really aim to maintain a healthy gut in terms of the gut microbiome to protect themselves or even prevent more severe disease. So with this microbiome immunity formula, during one of the waves of the COVID-19, we started doing a pilot clinical trial at our hospital. So half of the patients, 25, were randomized to have the microbiome immunity formula, and another half received just standard therapy when you are admitted for COVID-19. Bear in mind, every single COVID-19 cases in Hong Kong needs to be hospitalized in hospital, whether they're symptomatic or asymptomatic. And you could see here, they received 4 weeks of intervention. And we then look at the microbial assessment at week 5 and follow them up. What we found was that in the group who received this microbiome immunity formula, SIM01, at week 2 and week 2, significantly higher proportion actually achieved complete symptom resolution. And also more actually had antibody formation as well. We then look at the blood level and the pro-inflammatory cytokines. We also find that those who received the microbiome immunity formula SIM01 had lower or significantly lower pro-inflammatory cytokines in their blood samples following 4 weeks of treatment. And with that, I think it's quite clear that around the global view, it's well recognized that the intestinal microbiome may affect severity of COVID-19. And Fox News introduced some of our work in the United States and also in MedPage, explaining perhaps this is one factor explaining why some people are symptomatic, why some are not, why some are more severe, whereas others are not. And just recently, there's a case report published in Gut, whereby they have used fecal microbiota transplantation to treat patients with recurrent Clostridium difficile infection, which is now what's been done, for example, in the United States as well. And what's interesting is that 2 of these patients happen to be SARS-CoV-2 positive as well. And following the FMT treatment, the fever resolved very rapidly. And also the CRP resolved as well, and there was no significant adverse events. I think we need to keep an open mind, but at least for now, it just seems that certain form of microbiota modulation have not shown huge, safety signal, but may be beneficial, but clearly larger trials are needed to actually confirm that. So the last part I just want to highlight in 2 slides is this importance of vaccination. Global vaccination is occurring and it's really helping to reduce mortality and preventing transmission. But it's also very well known that our gut microbiota influence our vaccine response, and there are great work relating to vaccine, to influenza, to smallpox as well, showing how a well-balanced microbiota with high diversity clearly can actually impact on increased antigen-specific response. In contrast, if there are impact gut microbiome functions or immune function, it can lead to a blunted vaccine response, and whether this may actually be related to COVID-19 vaccine as well, especially we understand that durability of vaccine protection is one of the greatest unmet needs right now. So in a pilot study, we started looking at whether if you improve a gut microbiome, you can enhance the safety and efficacy of your vaccination. And we did some in vitro work and a pilot work, and we found that in this small group of subjects, they were able to enhance certain neutralizing antibody formation when we introduced a certain group of microbiota gut bacterial composition. And we just completed a clinical study, and a randomized control trial is now ongoing, looking at microbiota modulation to affect vaccine response and adverse effects. So the holy grail is, which is a chicken, which is the egg? Everyone will ask, is it because they have COVID-19, so they have dysbiosis, or is it the imbalance in the gut microbiome that lead to them having more severe COVID-19? I think there are now some science and some animal work to show us that indeed when you introduce a certain unfavorable bacteria composition, it can actually lead to more severe COVID-19. So there may be a component of causality in here. So in summary, looking about the importance of metagenomic sequencing, understanding gut microbiome, based on shotgun sequencing, we found that COVID-19 patients have very distinct gut microbiota composition when compared with non-COVID-19 controls. And this is concordant with the disease severity and also the concentrations of their inflammatory markers. They actually have depleted gut microbes that are known to have immunomodulatory potential. And some of these lasting compositional differences in the gut microbes go beyond clearance of the virus and may contribute to the so-called post-acute COVID syndrome. And one of the future potential is that by restoring our gut dysbiosis, this may serve as a complementary or an adjuvant treatment to hasten recovery for those who are infected, or to improve vaccine response to those who have received vaccine, to actually prolong the interval whereby we need to receive booster shots again. And many of this work were clearly conducted with the support of all our people behind the scenes, our postdoc, our scientists, and our assistant professors at the Chinese Students' Field of Hong Kong Microbiota team. And with that, thank you again for listening and for having me. And I'll be happy to take any questions.
Yeah, thank you, Prof. Ng. That was a fantastic and interesting talk. So we do have a few questions for you from the audience, and this is by Gabriel Yunn. So he's asking, do we know whether the gut microbiome from those with long COVID match with those from patients with other conditions, for example, the chronic fatigue syndrome?
That's a great question. I think that's the next thing that we really need to know. I mean, right now we haven't had a direct comparison. We've been focusing on the longitudinal cohort of from someone gets admitted to those who develop long COVID and those who do not, to understand the prediction of where this actually happened. And it seems like their baseline gut microbiome is really quite disturbed to start with. So the next question is whether this long COVID changes. And you saw that earlier on, I already were able to cluster some of these features to those predominantly fatigue, those predominant respiratory symptoms, and some with predominant neurological symptoms. So probably the next question is whether this multi-system disorder may mimic what we see with chronic fatigue syndrome following a certain infection. So we probably need to do some direct comparison for that. But also more importantly, to understand that how long I think this is going to last and whether there's something we could do after that.
Okay, that's fantastic. So you mentioned about that clustering. So just a follow-up on that question. Are you able to cluster with certain classes of metabolites? So I assume that your clustering is, organisms, but are you able to do that with metabolites?
So we started digging a little bit deeper on that, and we found that there are common pathways that are related to certain bacteria, which probably is not sort of surprising. And it seems like, for example, the isoleucine and the short-chain fatty acid or the butyrate pathways seems to stand out quite a lot. And we were able to see that clearly those who actually develop long COVID have certain that are more related to the Eubacterium, the Rosebaria, or the F-Prof sort of genus family. And these are the ones that produce a lot of the butyrate in order to defend against our epithelial cells or external pathogens. So the question is, like, you're absolutely right, it may not necessarily be the bacteria that we need to target, probably the downstream of what they actually produce, because the amino acids might be more easy to target, even if you want to think about small molecules to target for future therapies.
Thanks. So, yeah, there is another question about your SIM01. So the question is it proprietary? So I assume it must be. Yeah.
Yes, it is actually proprietary, and it's a combination of based on different types of sort of ratio. And I think what's intriguing is we did this in the Asian population because we understand that actually probably Asians' gut bacteria is very different. So when we look at 1,500 of the controls, we find that 40% of the Hong Kong citizens really are depleted in very certain very beneficial sort of bacteria. But this SIM01 is proprietary. It's got a sort of algorithm. It's a patent of the CUHK that has now been sort of commercialized actually for common use as well.
So, yeah, without revealing what it is, do you think that this works with inducing colonization, or it doesn't really colonize, but it affects the gut immune system?
It colonizes, because what happens is, and when we look at them in the fecal sample with shotgun metagenomic sequencing, kind of before and sort of after 4 weeks, we find that actually probably 70% of subjects actually had high abundance, so it actually sort of colonized in the gut. But what's intriguing is when they actually stop it for another week, actually they can decolonize as well. So the question is, how long may it take actually for it to actually remain sort of colonized? And I think we know that this may vary between different people as well.
Okay, just a last question from Siew Pang, and so she's asking, how long after taking the SIM101 will it restore the recipient's microbiota back to normal, and how long does the effect last?
At the moment, we see that probably at about 3 months, actually. So by taking that 3 months, you can maintain a sort of constant, sort of a more stable level for the majority of sort of people. Then how long I think it lasts, that really depends on what the function is. I mean, we had some early work now looking at sort of vaccine response as well. But for the COVID-19 sort of patients, we see that by just a month of sort of therapy, I think you can increase antibody and hasten sort of recovery. But it takes at least 3 months to colonize. So I would normally say about 3 to 6 months would be a sufficient period of time.
Okay, thank you very much, and all the best with your SIM101. So thank you very much for joining us today.
Thank you.